Hi everyone, my name is Kate Lynch. I'm here to discuss with you cystic fibrosis, a little bit about it, what causes it on the molecular level, some modes of inheritance, and some genetic testing that is available. So a little bit about what cystic fibrosis is. Genetics Home Reference describes it as the buildup of a thick and sticky mucus that causes damage to many of the body's organs. So the disorder is caused by a mutation in what is called the cystic fibrosis transmembrane conductance regulator gene. So that is a very big mouthful. So for now, we're going to call it the CFTR gene. So we're going to go over a lot of the details of the gene a little bit later. So for now, all you really need to know is that if there's a mutation in the CFTR gene, the mucus that is naturally produced by the body becomes very thick and sticky, and it coats vital organs of the body, including the lungs and the pancreas. So as a result of this buildup of mucus, the airways and the ducts of the body are clogged. And this is also a recurrent pattern. And this results in permanent damage of the lungs, as well as formation of scar tissue, which is also called fibrosis, and the buildup of cysts in the lungs, hence the name cystic fibrosis. So this mucus buildup also affects the pancreas, and the pancreas is an organ in the body that also produces mucus. So the pancreas's main function is to produce insulin as well as digestive enzymes, and when it's coated with this thick mucus, its functions are impaired, and this can lead to digestion problems, as well as cystic fibrosis-related diabetes because of the insulin production impairment. So over here we have a picture of a nice healthy airway with no cystic fibrosis, no mucus buildup, the air flows through nicely. And then over on the right, we have a cystic fibrosis afflicted airway. And we can see all this mucus on the sides and how air is really fine and small, and it's really hard for the person to breathe. Okay, so on a molecular level, now we're going to discuss more about the CFTR gene and what it does. So we know that a mutation of this gene causes the disorder, but why? So the gene's main purpose is to move negatively charged chloride ions out of the cell and into our body. So it's important to note that chloride makes up sodium chloride, also known as salt, and it is also really important to know that on a cellular level, water always will follow salt to maintain a level of equilibrium in the cells. So in turn, salt is kind of what controls where water goes in the body. So water also contributes to the mucus in our bodies, and it makes it thinner and kind of free-flowing. So when there's a mutation in the CFTR gene, the chloride channels that the gene codes for are inhibited, and the chloride or the salt doesn't move to where it's supposed to go. So this leads to water not also going where it's supposed to go. And then mucus isn't free-flowing or watered down anymore, and this produces kind of a thick, sticky mucus, and it coats the lungs, the pancreas, and other mucus-producing organs. And this buildup leads to the clogging of the airways and the ducts, and it causes the symptoms of the disease. So over here on the side, there's a really nice diagram of a healthy CFTR gene. So the individual has inherited 2 chromosomes from mom and dad that code for a nice healthy CFTR gene. So the CFTR gene codes for a CFTR chloride channel, and here at the number 3 we can see chloride is transported from the inside of the cell, out of the cell, and then these little hair-like structures are called cilia, which are outside of the cells, and they push mucus along to where it's supposed to go within the body. So these cilia are nice and lubricated with mucus that is nice and thin from the water, and everything is all good. So if an individual inherits 2 chromosomes from mom and dad that code for a mutated CFTR gene, the CFTR chloride channel is also inhibited, so something is wrong. So here the chloride transport isn't really working how it's supposed to. So the chloride is either not coming out at all, so these are not here, or there might be like 1 or 2 rather than these 3. So the cilia then build up all this mucus that isn't watered down, and it gets thick and sticky, and then it clogs the airway, and that produces the symptoms of cystic fibrosis. So how is this inherited? So it is inherited via the recessive trait from the parents, and it also needs to be homozygous recessive for it to express itself. So in other words, the child needs to inherit 2 recessive traits from mom and dad for it to express the trait. So mom and dad here are both carriers. The yellow is the recessive and the gray is the dominant. So we can see here child number 1 has received 2 grays and it is unaffected, which means it is homozygous dominant and it does not express the trait. The second and third child both get 1 recessive and 1 dominant, which means they are both carriers, but they will not express the trait because they have that dominant allele that masks the recessive. And then the last child will receive the recessive from father and recessive from mother, and this makes it homozygous recessive, which will express the cystic fibrosis trait and it will be afflicted with the disorder. So this is a very detailed pedigree, which shows the mode of inheritance throughout generations of the disorder. So these first 2 up here, Brad and Liz, are both heterozygous. Neither of them express the trait, and they are very similar to mom and dad in this picture. So there are carriers, but they do not express the trait. So we will look over here at this Punnett square that has both of their genotypes. Big A, little a, big A, little a, and we see that if there's a 25% chance that one child will have cystic fibrosis. So they have Chris and Brian and Olivia. So Brian and Chris neither are affected. One is homozygous dominant, no, not even a carrier of cystic fibrosis, and one is heterozygous, a carrier but not afflicted. Then the third ends up being the homozygous recessive, as we discussed before. Olivia is, in fact, afflicted with cystic fibrosis. Olivia then marries Mike. They have 3 kids, and because Olivia is homozygous with little a, and Mike is heterozygous, big A, little a, this is their Punnett square. Little a for Olivia, big A, little a for Mike. And then we can see that there is a 50-50 chance that their child will have cystic fibrosis. And we can see that Lizzie and Alana are both heterozygous, big A, little a. And then Daniel is afflicted with this cystic fibrosis because he is homozygous recessive as well. Similarly to Brad and Liz, Brian and Kelly have the same genotypes as Brad and Liz, and they will have the 25% chance of producing an offspring with cystic fibrosis. They have 2 children. One is heterozygous, big A, little a carrier but not afflicted. And then one ends up being one of the 25%, Lily, with little a, homozygous recessive, and she has cystic fibrosis. And then we come over here, Kelsey and Chris. Chris is homozygous, as I said before, dominant. Kelsey is heterozygous and big A, little a, and their Punnett square is down here, and we can see there's a 0% chance that they will produce offspring with the disorder. They do, however, produce an offspring that is a carrier. That is John, and he marries Holly, who is also heterozygous, and they will as well have this first Punnett square possibility of producing an offspring with cystic fibrosis, and they end up having Jen, who does have cystic fibrosis. Okay, so now that we know a little bit more about how it's inherited, we can discuss some genetic testing that is available for early diagnosis and treatment of cystic fibrosis. So the first step is cystic fibrosis testing of the parents to see if they are carriers of the disorder. This is called preconception carrier screening. If the test ends up coming back positive and both parents are carriers, the couple is then offered antenatal genetic testing. And this antenatal genetic testing is a procedure performed during the pregnancy to detect a health problem in the growing fetus. So they can test the child while it is still growing in the womb to see if the child will be afflicted with cystic fibrosis. The final genetic test is neonatal screening, and this is done after the infant is born. And they test for immunoreactive trypsin, which is in the plasma, and this is an indicator of a mutation of the CFTR gene. If they find that these levels are elevated, they will use a definitive diagnosis test, which is often a sweat test to measure any raised levels of chloride. All right, now you know a little bit more about cystic fibrosis. Here are some references for my information, and here are some references for the pictures.